And after a long session, probably I would like to start with a good news. The good news is that today people live longer. In this slide, you see life expectancy, average life expectancy in high-income countries during the last 350 years in Europe. Basically, 350 years ago, average life expectancy was approximately 35 years. Then, since 1840, it started to increase linearly. You see the curve is really pretty linear. And has reached today 85 years as a life expectancy. If you follow the dotted line, you see that if things don't change, in 60 years, people are expected to live, to have a life expectancy of 100 years. Now, this is a good news, especially for people of my age. They're getting to the place where, some time ago was a problem, and today you're still doing well. And it's a problem for our society because we have the aging society, all the health expenses all the budget are becoming a problem. And not too many people see this as an opportunity. I do. And you may ask, what's the connection between this and vaccines? Well, the connection is the following. Most vaccines have been developed in, Greg Poland said, in the post-war period. Basically, in this slide, I define the period when life expectancy used to be 60, 70 years. And therefore, the big need was in infants, a society with a lot of children, a lot of disease in children, and very few elderly. And vaccines, as we all know, have been tremendously successful in doing this. They've basically been able to eliminate most of the infant diseases and children diseases. And somehow they've done their job for that kind of society. The problem we have today is that our society is different. It's the one on the top. We have fewer children, a lot of elderly. So basically, the vaccines have been developed of a society of 50 years ago. Today, we have different society, different expectations, different medical needs, and probably even vaccines will need to ask different questions. So what can vaccines do for the 21st century society? And I believe this is what they can do. You see, different age groups. Vaccines so far have been focused mostly in infants and children. They've done a tremendous job. Most of the diseases are gone. We have a couple of them that we need to finish. One is Meningococcus, which is arriving. The other one is RSV. I will mention that later as well. But once we are done with those, with infants and children, probably we are done. Then, to the right, you have the elderly. That's the growing population. And that's where most of the budgets of all care go. And that's where, we are not doing very much. We have a a vaccine for influenza, developed a long time ago. We have zoster, maybe some pneumo now, but that's it. Really, we are not doing very much in the vaccine field for the major population as a big medical need today. So what about boosting this population with some of the vaccines for the children? They do get these diseases again. What about taking care of some of the problems of this population? Many of these people end up in the hospital for one reason. They die because of an antibiotic-resistant infection. Can we develop vaccines for antibiotic-resistant bacteria? Sure, we can today. And finally, cancer. Cancer is the disease for this population. It grows exponentially in this age group. And today we heard a lot, we are doing a lot to cure cancer. But vaccines so far have not been great, although they're improving in curing. They're great in prevention. can we prevent vaccines? Can we prevent cancer? Can we basically immunize people when they're 50 or 60 and delay prostate or breast or colon cancer by 10 years or 20 years? Or why not forever if we can? Now, all these things are becoming possible thanks to the technologies we have today. I could mention all the other age groups, but the only other one I want to mention is pregnancy. Basically, today, even with the best vaccines that we have, there's no way we can protect infants when they're 2, 3 months old from pertussis, from meningococcus, from RSV, and some of these have the highest mortality in this age group. So the only way we can prevent these diseases in the first few months of life is by immunizing pregnant women. Now, that was not possible because mostly of liability problems 10, 15 years ago. Today, thanks to influenza, thanks to the experience with neonatal tetanus, we know that it's possible to immunize pregnant women. The third trimester is safe and very effective because you can protect the mother and the child. So basically, there is a lot to do for vaccines for this society. And I'm not mentioning therapeutic vaccines that have been mentioned a lot during this session. So there's a lot we can do. And then there are a lot of other needs that we have today that you see in this slide. So basically, a lot needs to be done still with vaccines. And the question is, do we have the technologies to do all these things? Can we actually do these things? And here what you see is basically what has happened during the last 30 years. Vaccines up to 1980s were developed with the classical empirical approach that you see in the smaller circle at the bottom. And basically that's how most vaccines were developed. Then a number of waves of innovation occurred since the 1980s. The first one was recombinant DNA, the next was glycoconjugation, the next genomics, reverse vaccinology, and today we are living in a continuous wave of innovation that is really growing exponentially, and I think is going to change the field enormously. Now, this wave of innovation has changed a lot, has made possible many vaccines. For instance, recombinant DNA made possible hepatitis B and many other vaccines. Glycoconjugation made possible a lot of the successful vaccines we have today. Haemophilus, Meningococcus conjugates, pneumococcus, and others are in development. We didn't have the technology to do these things. I'll give some examples of reverse vaccinology, the new technologies. But I wanted to, before I go into that, I wanted to share with you this slide. Basically, in 1980s, barrier to vaccine development was mostly discovery. We did not have the science to do new vaccines. So discovery took a long time, sometimes forever, because we couldn't make vaccines. Development was easy. Hepatitis B was licensed in 600 subjects. Now, thanks to all these new technologies, discovery now has become much shorter and much more powerful because we can analyze and process a lot of data quickly and move fast. Development, however, hasn't changed at all. Actually, has not used the science to change the way we do development. And so basically the timelines for development, the cost, have increased enormously. And that's the biggest barrier we have today in vaccines development. Long times and very expensive developments. And the reason is that since we don't use the science, we collect few data in a lot of people and it takes forever. However, and I think Greg Pollan and the others this morning have shown you that today we can apply the science to development as well, collect a lot of data on a few people in a much shorter timelines, parallel trials, and shorter the time of vaccine development and cost. And that's the challenge that we have for the vaccine 2020 or 2.0, as Craig mentioned this earlier. Now I'll give you a few examples of the what the technologies did. One is reverse vaccinology. Basically, back in '95, there was no way, at least I couldn't see a way to develop a new vaccine for meningococcus B because all the technologies we had at that time were not working. Craig Venter published his first genome of a bacterium. I went to talk to him. We decided to collaborate to do the genome of meningococcus B. And from the genome, basically, we started to select by bioinformatics the antigens. We selected 600. We expressed them in E. coli 350. We immunized mice. We found 30 new antigens that people had not found, that were not going to find without the genome. Finally, we selected 3 of them. And we made a vaccine, which worked well in preclinical studies, went phase 1, phase 2, phase 3. And finally, basically in the fall of 2012, this was recommended by the European Medicinal Agency and finally approved in February this year by Europe. This vaccine has received a lot of press attention, especially in the UK, where there is a lot of need for this vaccine, but there's also Australia, many other European countries, there's a lot of expectation. And we hope that thanks to this new technology, we will be in the position, basically, to eliminate meningococcal meningitis from our planet. The reverse vaccinology has been pioneered by the meningococcus B obviously can be applied to many other bacteria. You see at the bottom many bacteria for which has been applied, but some of those which are really the challenges for today, like this antibiotic-resistant bacteria, can be approached by this. And in fact, there are, thanks to the genome, there are phase 1 or 2 studies of staph, E. coli, C. difficile, and Pseudomonas vaccines. So basically, all these vaccines are become impossible. The next innovation are adjuvants. From 1924 to 1997, the only adjuvant used were for humans have been Allen salts. In 1997, we licensed MF59, the all-water emulsion. And today, there are another 3 adjuvants licensed. And there is a parallel meeting on adjuvants going on right now. So I will not spend more time a lot of time on adjuvant. I just want to show the power of adjuvants. This is a phase 3 efficacy trial published in October 2011 in New England Journal of Medicine, comparing the efficacy of a licensed influenza vaccine in children from 6 months to 3 years of age. And in blue, you see the efficacy of vaccine without adjuvant, 43%. In red, you see the efficacy of the vaccine with the adjuvant, 86 to 90%. These are new tools that really can make a difference. And we know that this is the only example I want to show, but basically the same vaccine in the elderly has been shown to reduce hospitalization by 22%. In a Canadian study, to reduce morbidity by 25%. And all this, and today there are a lot of new adjuvants are being developed. So basically, I think the adjuvants will change the way we are making vaccines. And today there are a lot of small molecule adjuvants being developed as a TLR agonist that are also going to change it. But this is the only slide I want to show because you see the power of adding the right adjuvant to the right vaccine. One area that basically a new technology that is becoming powerful today is what I call structural vaccinology or structure-based antigen design. I think there's been a question before, when do we get a universal influenza? And I think the answer, I don't have an answer to that, is not soon, but basically, the reason why we get the question, why do we get when do we get a universal influenza vaccine, is shown in this slide. Basically, this slide shows the influenza hemagglutinin is a trimer, and on this trimer, all the protective antibodies bind. On the top or the head, the antibodies that we know that protect from each different variant of influenza. In the stem, in the lower part, you see the new antibodies have been described during the last 3 to 5 years. These are human antibodies that have been selected, that basically they are able to neutralize most and sometimes all influenza viruses. And these antibodies have been published in many top journals, Science, Nature, all the top journals. And these antibodies are probably good for therapy because they're human antibodies, they work very well. All these papers, in the conclusions, they say, well, we have found a universal epitope for influenza, and therefore now we're going to have universal vaccine. Now, all the rest of the paper is true. This last sentence is just wrong. The reason is that we don't know, once we have an epitope, how to transform that epitope into a vaccine. And we are improving our knowledge. Maybe in a few years we're going to get there, but today we don't know how to do it. The reason, I'll give you an example. This is an epitope of one of the antigens of Meningococcus. If you characterize this epitope by Western blot, you will find the blue is the epitope. If you use phage display library, this is the epitope. If you use peptides, this is the epitope. If you use site-directed mutagenesis, this becomes the epitope. If you use deuterium exchange, now the epitope looks different. If you use X-ray crystallography, you will find that it is much larger. And this will be the final epitope. So basically, the way we define an epitope depends a lot on the method that we use to define the epitope. So basically, we try to make an epitope for a B cell depending on the technology that we use, but that's not the way. So basically, we use different glasses to see an epitope, but we don't know which glasses the immune system uses, and that's our problem. And so until we solve this problem, we'll not be able to make a universal vaccine, at least with that epitope. But if you understand this, basically you can actually treat the epitope not as a 1 or 2 amino acids, but as a surface. If you work as a surface, then you can start to engineer. And here is a paper that we published where basically we've been able to engineer the surface, not the epitope, the area, or epitopic area, and transform 3 variants of immunogens into 1. So basically, it's a promising field, but still we don't know how to solve it. Another example of structure-based design or structural vaccinology is RSV. A few years ago, we didn't have the structure of the RSV trimer, which is the vaccine antigen, but we knew that it had 2 states, the pre-fusion on the left and the post-fusion on the right from the structure of the parainfluenza virus. And we knew that the natural viral protein purified for the virus is so unstable going into the 2 structures that you cannot make a vaccine. So we have been working, and many others, trying to stabilize one of the 2 structures. The preferred one will be the one on the left, the pre-fusion. We have been so far successful in stabilizing the one on the right, the post-fusion, and that has been knowing the structure, just deleting few amino acids, the fusion peptides. So we've been able to make a molecule now that can be stable, uniform, can be manufactured, it can go into, is protective in the animal models, inducing neutralizing antibodies, it can go into trials. So structural vaccinology is starting to help, is making a lot of progress, is not yet there to make the universal vaccine. Wanted to mention another technology, what we call SAM. SAM stands for self-amplifying RNA. This technology came from the frustration of working with viral vectors. As you know, many are working with adenovectors, MVA, and other vectors. We have been working for many years with alpha virus. And we're trying to make a packaging cell line that will comply with all the regulatory requirements to be safe and approvable for use in humans. And while we could get very good packaging cell lines in the lab, packaging cell lines that will meet the regulatory requirements have been a frustration. So to the point that one day we said, why should we actually package the RNA into a virus? Why don't we use the RNA as such? So basically, we took advantage of our colleagues that have been doing delivery of RNA using nanoparticles for siRNAs. We took their delivery nanoparticles, we mixed it with our RNA, alpha virus RNA, and we found that basically the RNA delivered by nanoparticles was as good as the viral vector, as good as DNA electroporation in inducing antibodies against RSV. We looked at neutralizing antibodies, and we found was as good as the viral vector, and also in reducing the plaque-forming units of the virus. So RNA was looking pretty promising, induces CD4 responses on the left and CD8 on the right. So basically, today, this is a new technology. It works beautifully in all the animals we tested, and we are trying to confirm whether it's going to work as well in humans. The beauty of that will be that, today vaccines are biologicals. You use a fermenter on the right. To make RNA vaccines, the SAM vaccines basically use a tube with chemicals and enzymes. So we'll go away from big manufacturing plants, and you will go into, probably step into synthetic vaccines. We are moving towards phase 1. Once we get the results from phase 1, I'll tell you whether they work in humans, but so far they're very promising. The new technologies are changing enormously the way we do vaccines, and in Europe, actually, I'm coordinating one program called Aditech that is basically funded just to develop the new technologies. And I think influenza has been mentioned many times today, and the question is, can we innovate in influenza? At the end, influenza, we started making vaccines in eggs in the 1940s. Well, a little bit of evolution from all virus to split and subunit in the 1970s, but we have really been moving very little since then. And the main question is that influenza, should we continue with eggs forever? At the end, they've been working well for a century. We recently got a cell culture vaccine licensed. So where should we go in the future? And usually, if there is no difference between vaccines, at the end, you don't care which technology you use to make the vaccine. But there is a difference. And the technology, when a technology becomes a limit to the advancement of the science, then is the time to start to retire the technology. And to explain that, look at this slide. This is taken from New England Journal of Medicine, February 13 of 2013. And also on the CDC website. And basically shows the influenza seasons from 2000 to today. And you can see below the dotted line that most of the seasons were relatively normal. But there have been 4 peaks of abnormal severity, mortality, and things. Which are these 4 peaks? The first one happens very early in September, October, week 40, was the 2009 pandemic. Sure, that was unexpected. People were not immune to that. The vaccine came late, so we got a peak of pandemic. What about the other 3? This one is 2003-4, and is I don't know how many of you remember the Fujian strain. The vaccine had been made with one strain, but the circulating strain was a Fujian. And basically, it was mismatch between the vaccine and the circulating strain. It was a lot of disease. What about the other one? This one is 2007-8 season. Again, there was a mismatch between the vaccine strain and the circulating strain. And what about this one? Well, this is this season. It's the 2012-13 season. And you know there's been a lot of disease, a lot of Boston, New York was a very high incidence. All the emergency rooms full of, and what was the reason? This was a season where there was no mismatch between the vaccine strain and the circulating strain. At least that's what people thought. In fact, there was a mismatch. And the mismatch was that between the the vaccine produced in eggs and the circulating strain. So the strain had been selected, growing in cell culture, was the right one. But when it was passed in eggs, basically acquired mutations, and now was very good against the egg-grown virus, but was not good at all against the circulating strain. So this time, the work had been done correctly, but there was still a mismatch. So the conclusion is that 3 out of 3 times, except the pandemic, when we had a high incidence and severity of influenza, has been caused by vaccine mismatch. And why do we have vaccine mismatch? Many reasons. But you see, in this slide, you see when the centers try to adapt circulating strains from cells, like MDCKs, into eggs, you see how many strains of the selected strains grow in eggs, 1%, 2%. So the selection that you have in eggs is so limited that it's much easier to have a mismatch than if you grew in cells. And that's basically, on top of that, anytime you go from cells to eggs, you accumulate mutations. So basically, I think this is a reason why we should look more and more into cells, because the eggs, they do cause mismatches from time to time, and when that arrives, we have an increased severity and mortality from influenza. Now, can we go beyond that? Today, to produce a vaccine seed, you know, there are the WHO collaborating center, they send the viruses to CDC, CSL, NIBSC, and then they do reassortance, and then eventually the vaccine seeds are sent to the manufacturers. Collaborating with Craig Venter doing synthetic biology, basically, we developed a technology where basically we get the sequence on the internet as soon as a new virus is sequenced, and we synthesize the gene, and in 5 days we have the seed to produce the vaccine. It's a new technology, can be useful or not, we'll see in the future, but it's very promising. I mean, what we see is that the future could go from basically a lot of work sending the viruses around through customs, through things, all the problems that we have, through basically an internet like vaccines, where basically you ship the sequences, you ship just the information, you don't ship any biological samples. The information is collected, it's processed. You could get automatic sequencer completely free of humans that basically could make the vaccine, if it's made by RNA, basically could be done entirely without any human basically intervention. It's maybe science fiction, but not too much, because 50–60% of that we can do already. So I'm not sure whether this will be applied, whether it will become a product, but this basically wanted to show that technology is moving, we need to think differently, and technology is going to surprise us continuously every day. I think I'm getting to the end, but before I do that, I think we should never forget that in addition to looking at the future, we also need to look around. And today we know that we have all these beautiful technologies we can apply for us, but there are a lot of these technologies could be already useful in developing vaccines for which there is a medical need but no market. And therefore, I think it's important that we think also making vaccines for poverty. And here I'm pretty proud to say that Novartis has been able to set up an institute that basically is completely devoted to use these technologies to develop those vaccines for which there is no market, but there is a lot of medical need. And with that, I would like to thank you for your attention.